All right, let's get right to it today. Just a quick reminder, remember noon on Monday. So don't come at 1, eat lunch a little early before you go. Come at noon. Also, homework due Monday at noon as well. So when I kind of checked to see how many people were doing it, it was kind of low the other day. So just don't forget about those points. So I probably should let you know how many points you have, but there's not an easy way for me to do that. Some of you may not need to do all the homework and you've got enough to still get full points. But, okay, so the plan today, get through the rest of our new material. I'm gonna kind of trim back some of the COVID stuff to historical stuff so we don't get bogged down in that, but we gotta finish HIV. And there's really just kind of a few things I wanna mention about COVID. And then we'll do a little bit of reviewing so we can kind of go a few different directions with that. Last time looking at the structure of the HIV, it's important we'll think about kind of how the life cycle of the virion in a minute. So some key things about the structure of that virus, the three enzymes, the three proteins that it brings with it, they're all enzymes: reverse transcriptase, integrase, and protease. And then some key spikes on the outside of the virus, the GP120 and the GP41. So let's see how those all come into play when the virion is infecting a cell. HIV will infect cells that have those CD4, that cluster of differentiation on its surface. Now, the most famous kind of cell that has those, remember, T helper cells. But actually, there are other types of cells that have those CD4 glycoproteins, macrophages. What are some other ones? B cells, even some granulocytes. And we don't really talk about that very much. HIV, even though it infects those other types of cells, it doesn't seem to damage them very much. So it's the T helper cells that really get decimated with this infection. So that's kind of the ones that we talk about. Okay, so that's GP120, that's the kind of the little red, the way it's drawn on here, it's not drawn as a spike, but that's the spike, we'll call it, on the HIV that recognizes the CD4 that's important for attachments. And that also kind of tricks the outside of the host cell into endocytizing the virion. So entry by endocytosis, that's kind of forms a membrane-bound sac around the virion. And then the GP41 is somehow involved in the uncoating process. So getting rid of that endosome membrane, the membrane around the envelope around the virion comes off, as well as the capsid breaks open. So what comes out are those three proteins, maybe some other stuff that we're going to talk about, as well as the nucleic acid, that positive stranded, single-stranded RNA. All right, so right away, that reverse transcriptase, so it talks about right here, goes to work. And that, remember what that does, that makes the single-stranded RNA into double-stranded DNA. The double-stranded DNA will enter into the nucleus of the host cell and will integrate with the help of that enzyme integrase into the host DNA. Okay, so then we would call it a provirus when that nucleic acid is integrated into host DNA. It doesn't ever come out. Okay, so we've seen that latency in animal viruses. There's no induction. Okay, as far as we know, not a way to get it out again. The virion will eventually start transcribing and translating its RNA into viral proteins, viral parts, the assembly process into new virions. Eventually that new virion is released. And then once that release happens, interestingly enough, that's where the protease functions. There's some cuts that need to be made in certain proteins to make them functional. So without those cuts happening, the virion is not able to replicate itself later. So we won't get any more specific than that, but okay. So those three enzymes, those are unique to the virion. So that makes a great drug target. All right, so it seems reasonable to talk about treatment here as we're mentioning those enzymes. When we talk about treatment for HIV, we usually refer to that as, I think an acronym that they use. ART, okay, antiretroviral treatment. Okay, HIV patients have to be on a lot of medication in order to keep the replication of that virus down. Usually patients are taking at least 3 different drugs. These are the most common mechanisms of those drugs. They're going to target one of those 3 different enzymes. About half of the drugs that we have in that antiretroviral therapy are to the reverse transcriptase. Some of them are nucleoside analogs. Remember, what other drug have we talked about that's been a nucleoside analog? It's the one that we use to treat herpes. Acyclovir, some of you said it, right? Okay, so it's another nucleoside analog. So the first drug that we had to treat HIV was called AZT, azithromycin. And that drug is still used. They call it something different now. But this is a structure here. It's got the base that's really close to the structure of thymine. And then this is similar to deoxyribose, except instead of this 3' hydroxyl group that's normally there, we have an azide, the triple bonded nitrogen structure there. So anyway, that's close enough to the structure of a nucleoside that's the reverse transcriptase, which is going to make a lot more mistakes than our DNA polymerase 3, for example. That will get incorporated into the DNA as it's being replicated. And because there's not a 3' hydroxyl group, that will stop replication. No more can be nucleotides can be added onto us there. There are some non-nucleoside drugs that basically they bind to reverse transcriptase and then keep it from working. So a little different mechanism there. Of course, integrase inhibitors keep the HIV DNA from integrating into our host chromosomes. Protease inhibitors keep that protease enzyme from working so that the new virion doesn't mature and it's not able to replicate. And there are a few other mechanisms as well. But again, people have to stay on these drugs. There are some side effects associated with them. Some of the most common ones, nausea, fever, tiredness. Usually the side effects are worst when a person first starts taking it for the first few weeks. If the side effects go on and on, usually there's different drugs that can be tried to see if they cause less side effects. There can be long-term side effects from the drugs as well. And if you look at the list of them, it would fill up this entire page of the possible complications or side effects from the drugs. But generally speaking, if a person stays on their ART drugs, then they can live a normal lifespan. So we've made huge progress in this. In 1976, for a 20-year-old that was diagnosed as HIV positive, they were expected to live no more than 10 years. Okay, so again, today, normal lifespan as long as people stay on their drugs. They cost, the last time I checked, about $20,000 a year. So it's not nothing, but we are making some progress there. Transmission. all right, by very specific ways, direct and specific routes. I can remember I was a kid during the AIDS epidemic, the early days, and I can remember being worried that you could get AIDS from going to the swimming pool or sitting on a public toilet seat. Through bodily fluids, so blood transfusions, sexual intercourse, IV drug users, sharing needles, mother to child transmission. About a third of the time, an HIV positive mother will pass the virus to their child. So not maybe as often as you might have thought. HIV is really wimpy in the environment. So we don't get any fomite transmission because, again, in the environment, the envelope degrades really quickly. You can't get it from rubbing your nose and touching a doorknob or something like that, or by eating it even. Okay, so I looked this up the other day. It's still considered a pandemic. This is not gone away. Certainly, people are living longer with it, but there's still, as of just a few years ago, over 1 million people in the US that have this. It is still out there and being transmitted. There was a special on last week that I caught the end of it. There is medication that people can take if their partner is HIV positive, so that they do not contract the disease. So there's medication you can take to keep you from getting it. It's called PrEP, I think. But again, in 2011, the life expectancy was 70 years, but now they're saying it's normal. So whatever that is, mid 70s, something like that. Okay. Let's see. Questions. Oh, forgot one other thing to talk about. Your OpenStax presentation of kind of what's going on in the body, the graph that they give you is a little bit different than this, but it's similar enough. I kind of like this one a little bit better. It's a complicated graph, but it kind of helps you understand what's going on in terms of what kind of cells that it's impacting in the host. So when somebody is first exposed to HIV or infected, there's typically a pretty rapid increase in viral titers in the bloodstream. We do initially get some innate and adaptive immune response to that. So if you look at the purple line as the antibody, we do get some antibodies, and the HIV typically, it peaks and it comes down for a while, and will typically be latent for some period of time that's variable depending on the individual. But over time, eventually the virus will start to replicate. And as it's replicating, our green line here is the CD4 helper T cells. Those helper T cells are over time going to diminish. And of course, those cells are vitally important to activate not only our cytotoxic T cells and cell-mediated immunity, but also our B cells to activate them to make antibodies. So our ability to have an adaptive immune response diminishes as those CD4 helper cells are dying. A person is considered to have full-blown AIDS if they have less than, I think it's 200 cells per mil of CD4 helper cells. So I think that's a homework question, so I'll save you from looking it up. So over time, it's not the amount of virus in a person's blood that kills somebody with HIV. It's the fact that they are not having any immune response to the things that we're bombarded with every day. So what causes death are those opportunistic diseases, most likely. And there's diseases that you don't normally hear about people getting. This is not a list to memorize, but according to CDC, this is a list of infections that if somebody has one of these opportunistic infections and is HIV positive, then they're considered to have AIDS no matter what their CD4 count is. Question? Oh, 200 cells per mil, I think. I'm not sure about my units, or cubic millimeter of blood. I think that's the same thing. Yeah. Okay, so when you look at this list, the only thing that we've talked about in class is TB. Of course, that's a true pathogen. We often see that, especially sub-Saharan Africa, population of people that are HIV positive there and have tuberculosis is really high. But a lot of these things, again, they're opportunistic infections. You don't normally have them if you have a functioning immune system. So these are the things that usually causes somebody with AIDS to pass. All right, any questions? HIV, okay, remember it's a negative RNA, negative single-stranded RNA virus. No, positive, I get that mixed up. Positive, it's the flu that's the negative single-stranded RNA virus. All right, so one more virus to talk about, Coronaviridae, the family of coronaviruses. Up until a few years ago, I wouldn't have had a lot to say about this virus. It's a common cause of colds. A lot of different things cause colds, like rhinovirus, second most common cause of colds. There are some clinically very serious infections that are caused in addition to COVID-19. SARS-CoV, that stands for severe acute respiratory syndrome. We'll talk a bit more about that. And MERS, that stands for Middle East respiratory syndrome. Okay, so we'll mention those briefly. And of course, SARS-CoV-2, we named that virus SARS-CoV-2 because when we sequenced the DNA of from the gene that makes the RNA-dependent RNA polymerase, a really important enzyme, it was very similar to the SARS-CoV, so that's why we called it SARS-CoV-2 instead of MERS-CoV-2. And of course, that causes the infection, that virus caused the infection known as COVID-19. All right. It's a positive sense single-stranded RNA. So again, try to keep that straight, positive, negative, whatever. And it's like our other viruses that we've talked about, it's enveloped. Okay. A few more pictures of the virion. I kind of like this one on the left here. It's from the New England Journal of Medicine, and it's the original SARS-CoV virus. It's not SARS-CoV-2, but it shows the genome here. That's what this kind of long line with looks like little beads on it or something. That is the unsegmented genome. So remember, influenza virus had like 7 different segments. This is just one long string of RNA. It is the longest genome or largest, either way, of any RNA virus. And it encodes for 29 different proteins. So remember, we said smallpox was one of the biggest viruses, almost 200 genes. Herpes virus, still another big one, 80 to 100. Those were DNA viruses. Some RNA viruses are as small as only carrying 4 genes. But so this is one of the longest RNA virus. These little red, like I said, they look like beads. That's supposed to represent the protein capsomeres that make up a helical capsid to protect that genome. And then structural proteins on the outside, we could get into a little bit more depth here, but of course, there are spike proteins. That's the one, that's the most antigenic part of the virion. That's what we've got all our vaccines trying to target. The spiked proteins, they recognize the ACE2 receptors. So that stands for angiotensin converting enzyme 2. Okay, we have these receptors in our nose and our upper respiratory tract, and they're important to regulate blood pressure. So that's what COVID recognizes. And the other structural protein I'll mention here, there's an envelope protein as well that is most important for release of the new virion. So the spike proteins and the envelope protein, they're sort of analogous in a way to the two proteins that we talked about for influenza. Hemagglutinin to recognize host receptors, and then that neuraminidase that was important as the new virus leaves the host cells. All right, so little brief history lesson about SARS-CoV, severe acute respiratory syndrome. That burst out on the scene in 2002 and 2003. There were about 8,000 cases. And this is infection that is not circulating anymore. Okay, so no new cases since 2003. Had a little bit higher mortality rate than COVID, about 9%. This infection, there weren't really mild cases. You got it, you were really, really sick. Some of the same symptoms as what we see with COVID. People died just from respiratory failure. So that's something that actually helped us kind of clamp down and kept the disease from spreading, because if you got it, you were really sick. You weren't walking around asymptomatic giving it to other people. So they were able, again, with stringent public health mitigation to keep this from spreading and not have it spreading anymore. Hoping that would have been the case for COVID-19, but it wasn't. They believe all coronaviruses are zoonotic, meaning that there are coronaviruses in lots of different animals. Usually, though, they don't cross species barrier. So it's kind of a rare event, what we might refer to as a spillover, when now a virus that, say, was in bats is now suddenly able to infect other kinds of animals. So we think there was at least one spillover event. We've traced the phylogeny, the evolutionary trail of the virus back to this, I think it's called a lesser horseshoe bat of SARS-CoV. It's just a little tiny bat that would fit in the palm of your hand. They also call the civet cat a secondary host. And that just means that the virus gets kind of amplified in the civet cat. And you think that would be an odd thing for people to be around, but in China, it's in this part of southern China, it's considered a status symbol to eat like rare and exotic things. So that's the whole concept of the wet market, where there's all these different animals from all over the world and kind of all mixing around, and that can be a bad recipe for some zoonotic infection. Okay, so that's just a little bit about SARS. No vaccine, no treatment really. We haven't seen it since 2003. An infection that is ongoing is MERS, Middle East Respiratory Syndrome. That first emerged in 2012. Most of the cases are in Saudi Arabia because people that get it typically get it in association with dromedary camels. Okay, so a little camel lesson. The one hump is the dromedary camels that we find in the Middle East. The other kind of camel, the two humps, that's a Bassetran camel. That's mostly in Mongolia, Central Asia. Not many of those left in the wild. So people that get this respiratory infection almost always have been in contact with these dromedary camels. It can be transmitted person to person, but most likely from a camel. Pretty high case fatality rate, 35%. That might not be totally accurate because sometimes this manifests as a milder disease and it might not get counted. But when we know somebody has this, it's pretty severe. Again, we think originally there was some kind of bat was the original host, but now mostly people get it when they're contact. Okay, so people contract, I said this before, when in close contact with camels. Okay, so what do we do? Anybody that's, is around camels a lot, they have to wear personal protective equipment. They kind of pass laws to, kind of keep track of where camels are moving. They pass laws about drinking unpasteurized camel milk, and they pass laws about drinking camel urine. Apparently that's a thing, and a cultural thing. So there's a little article that talks about that. That is a no-no if you want to stay healthy. So yeah, I don't know. You learn something new every day, right? Same kinds of symptoms. Again, respiratory symptoms, a little kidney involvement, sometimes pericardium, lining of the heart affected. No vaccine, although it seems to me all we need to do is get a decent vaccine in camels and we could really nip this in the bud. So before class, I was Googling that a little bit harder, and there is a camel vaccine in progress, and they're hoping that will come. If we could, nip it in camels, then we wouldn't have it in people anymore. So okay, promising results, showing promising results. So hopefully we'll get to that. Okay, so that brings us to where we're at with COVID-19. Everybody's sick of hearing about this. The signs and symptoms. The Omicron variants are really having less loss of taste and smell. So we're not seeing that so much anymore. Deaths, I update this every time. I've taught this class. We're over 1 million now. The symptoms: tiredness, coughing, sore throat, transmitted by respiratory droplets. So wearing a mask really cuts down on the transmission of that quite a bit. Has the potential to be airborne, has the potential to be passed on fomites, but that's pretty not very common, we think. Let's talk about treatment though. We've already talked about the vaccine. So I won't go into that. You are considered up to date on your vaccination if you've had a bivalent dose. So that means a bivalent vaccine has had, you'll have memory for that original strain of coronavirus, as well as the new variants, the BA4 and BA5 Omicron. So that came out in the fall, early fall. So if you've had that, you're totally up to date. So what do we do to treat COVID-19? Supplemental oxygen, monoclonal antibodies, those are actually made in the lab and can be administered by IV. Usually the earlier in an infection that they're given, the better. Harvesting plasma from people that have recovered from COVID-19. What I was just reading before class, they said they're not really sure how much that helps. It doesn't do any harm, but they're really not sure how effective that has been. There's been a few drugs, one in particular that shows a lot of promise. So there's a couple of nucleoside analogs, and nucleoside analogs have the potential to be good for lots of different viral infections, to be more broad spectrum. This earlier one that they came out with, remdesivir, they're not really using that very much anymore. It's not terribly effective. Molnupiravir works a little bit different way. They were really excited about this. It came out a year ago. Remdesivir is given by IV and molnupiravir is given by a pill. So they were really excited about first oral medication. This is actually not a chain termination analog. It actually works by we'll just say the buildup of mutations, because it actually, the nucleoside analog will incorporate into the long chain of DNA or RNA, but then when it's, when the RNA is transcribed or replicated, it makes mistakes because it doesn't bind with the right complementary base. So we get this buildup of mutations, catastrophic mutations, they call it. So there are some worries about how safe that is. You would not want to give a kind of a medication that caused mutagens to somebody that was pregnant, for sure. So any tissue that's rapidly dividing could have mutations in it. So that never really gained traction, because at the same time, another drug came out that has a different mechanism, and that drug is Paxlovid. This is a protease inhibitor. Okay, so we just talked about protease in HIV. COVID-19 also has a couple of different proteases that it either brings with it in the virion, or it's one of the first few genes that are transcribed. Because when it makes this functional RNA-dependent RNA polymerase so that it can copy its genome, there actually has to be some cuts made in that enzyme to make it functional. So this animation here that's put out by Pfizer actually does a really nice job of explaining how this drug works. This RNA-dependent RNA polymerase is actually just kind of a bunch of kind of segments of amino acids, again, that has to be cut by this protease. So we have this drug that inhibits that protease enzyme, and it's very effective in slowing down the replication of the virion. So the statistics that I read, it's like 85% effective against severe COVID. Okay, so, and again, we've had this for about a year. When my son had COVID in early fall, I called the doctor. I said, can he take Paxlovid? And they didn't want to give it to anybody that wasn't in a high risk category. So it may be eventually anybody that tests positive goes on Paxlovid. I don't know. But with a combination of good testing and knowing that you're ill and drugs to treat it and being vaccinated to try to prevent it, you know, hopefully the whole pandemic, or I guess they're not calling it a pandemic anymore, but hopefully it will kind of fade off into the sunset and not be a problem anymore. Okay, questions? Any questions about COVID there? All right, so I wanted to fill up the rest of our time just reviewing a little bit. So the three things that I thought of that maybe we could best use our time is reviewing metabolism, reviewing some genetics, and reviewing just kind of what the different microbes are. So I just want to, just talk about the different microbes just briefly, because this is kind of basic stuff. And I don't want you to miss anything because you just kind of forgot something easy. I have a review that I have recorded of metabolism and replication and transcription and translation that I will post if I don't get to reviewing that stuff. All right, so unless anybody has any burning questions that they kind of would rather that I address first. Okay, all right, so remember our taxonomy. All right, well, first of all, I should say, make sure you're studying the most recent pathogens that we've done since the last exam. Spend most of your time studying that, okay? And then as you have time to go back and review some of these big picture things, remember all of life, we split into 3 domains of life. And that was due to the work of Carl Woese. And what did he use to split up his domains? He sequenced, okay, ribosomal RNA, all right, and he expected only to find 2 big categories of life, and he actually found that there was really 3 different distinct groups, and he called those the bacteria, the archaea, and the eukarya. Okay, so what makes those distinct? The bacteria and the archaea, remember, those are considered prokaryotes. Something that's prokaryote, it just means it doesn't have a membrane-bound nucleus. Okay, and it doesn't have membrane-bound organelles. And of course, eukarya, that's our eukaryotes. So we spend most of our time this semester talking about bacteria. How are they different than archaea? There's lots of little tiny differences. The main thing that we talked about is that the archaea do not have peptidoglycan, PG, peptidoglycan in their cell walls. Okay, so that's a really distinct thing about bacteria. If they have a cell wall, which most of them do, they have peptidoglycan in them. Peptidoglycan. Gram positives, lots more peptidoglycan in their cell wall than gram negatives, but they both have it. And then the eukaryotes that we talked about, of course, we talked about fungi, we talked about protists kind of briefly, plants, animals, okay, those are our eukarya. The difference between a fungi and a plant, fungi don't photosynthesize. Okay, protists, they don't have a cell wall. They have kind of a, most of the time they have a little proteinaceous layer that they call a pellicle. Okay, but they don't have a cell wall. Okay, so lots of eukaryotes do have a cell wall, not animal cells. Okay, so and then of course the other microbes that we talked about viruses, we talked about, these aren't considered in that tree of life. We talked about prions. Okay, viruses, we just got done talking about that. They got nucleic acid covered in a capsid. Prions are infectious proteins. Okay, so remember, they do not have DNA or RNA. Okay, and they replicate just by causing that infectious proteins causes other proteins to misfold. Okay, so prion disease, always fatal. Okay, let's do a little overview of metabolism. I think that's probably the thing that will help you most. And hopefully you can read my writing here. Stop me by all means if you have questions. So usually we start with glucose, and that's splitting of the glucose into the 2,3 carbon pyruvates. We call that, it's going to happen by some kind of a glycolytic pathway. The glycolysis is typically known as that Embden-Meyerhoff-Parnas pathway, or we'll call it the EMP. Okay, that's typically what people will call glycolysis, where you are inputting a couple of ATP and getting out more ATP by substrate level phosphorylation. And the other thing that we gain from that process is NAD gets reduced to NADH, okay, during that process. So those NADHs, if it's going to be, if the organism is doing some kind of respiration, that's going to convert into ATP, those electrons. But there are some alternatives to the glycolysis. In bacteria, they can do the Entner-Doudoroff pathway, and that's only bacteria. It's usually bacteria that are deficient in one of the glycolytic enzymes. They get a little less ATP. Again, that's only bacteria. And then the pentose phosphate pathway, lots of different organisms do that, but it's more so when they need to synthesize molecules rather than needing ATP. Okay, so those are the two other glycolytic pathways we talked about. Let's assume the organism has a suitable electron acceptor. Excuse me. that pyruvate, there's a little transformation step there, but the 2-carbon acetyl-CoA enters into the Krebs cycle. Off of the Krebs cycle, we get lots and lots of NADHs, lots of electrons being carried away, some by that FADH carrier. And those electrons are going to get dropped off at some kind of electron transport chain or electron transport system, okay? Where a series of carriers will pass those electrons, and the protons will get shunted outside of the cell. And you'll have some kind of electron acceptor here at the end. In aerobic respiration, that's going to be oxygen. But remember, lots of bacteria can use other things instead of oxygen as final electron acceptor, things like nitrate or sulfate, for example, some inorganic ion usually. Okay, so we call this a proton motive force, all those protons building up outside of the cell. I didn't leave myself enough room here, but there's one spot in the cell membrane where those protons can diffuse in, and that's at that enzyme called ATP synthetase. Okay, where those protons will diffuse in, and that powers the phosphorylation of ADP to ATP. Okay, so lots and lots of ATPs gained from aerobic respiration, a lot from anaerobic respiration as well, not quite as much. What if there is not a suitable electron acceptor down here at the bottom? Then the organism has no choice but to do some fermentative pathway, because if these NADHs that coming off of glycolysis have nowhere to go, then they can't recycle, the NADH can't recycle back as NAD, and then glycolysis will stop. Okay, for every oxidation reaction, you have a reduction, and if there's no reduction, you can't have the oxidation. So these electrons have to have somewhere to dump off. In a fermentative pathway, they will just dump off maybe some organic compound that's already in the cell, like pyruvate, for example. Okay, and then you would end up maybe with something like lactic acid, for example. Lactic acid is just pyruvate with that hydride ion with the 2 electrons and the proton attached to it. Okay, so in a fermentative pathway, that's just keeping those NADs recycling back. For bacteria, they're going to have some acidic end product. When yeast, there's many different fermentations, right? If you're in lab, you tested for a mixed acid fermentation or a 2,3-butane-diol fermentation. That can help us identify organisms based on what pathway they use and what end products we find. Yeast will do an alcoholic fermentation. Okay, most bacteria, they'll end up with acidic end products. So, and little to no additional ATP in any kind of fermentative pathway in addition to glycolysis. You could consider glycolysis as part of the fermentation pathway. Okay, but again, at least the organism still gets a few ATPs from glycolysis. Okay, that's my big kind of spew on metabolism. Any other questions there? Yes. Specific, do you need to know specific final electron acceptors? A lot of times it's going to be oxygen or some inorganic ion. So I don't know. That's probably as specific as you would need to know. Yeah. Yeah, now you're not trying to pin me down about what I'm going to ask and what I'm not. I think this will help you kind of knowing the flow of things. is where I'm most likely to ask a question about. Yes, I try to keep things a little bit more general. There are a few questions on the final that we consider assessment questions that everybody in the course gets asked. So there are some questions that I didn't write, but I did approve at one point. So, okay. All right, let me talk through at least replication. So let's remind you of the enzymes involved in replication with the time we have left here. So remember, replication is when DNA is copied from DNA, when we make RNA from DNA, that's called transcription, and then we make proteins from that RNA, that's called translation. So don't forget the basics. Replication, remember where that starts at a place called the origin. Okay, and in bacteria, all the DNA just gets copied. So there's not a start and a stop so much. You don't have to worry about initiation and termination like you do with transcription and translation. You just start at one spot and it all gets copied. So let's see, let's draw it like this. We usually draw it the other direction, so let's draw it this way this time. All right, so here's a replication fork. So and of course there would be another one going off this way, all right, because it's gonna, the circular DNA, and it's gonna open up. The main enzyme involved here, let's draw it like this, remember, is DNA polymerase 3. Okay, and that enzyme can only move in one direction. So we can't have it move both strands being copied at the same time going towards the replication fork. One's going to be going towards the replication fork. Let's see, I'll draw that here on the bottom. And one has to be kind of copied in a series of fragments that we call Okazaki fragments. RNA polymerase can't start from nothing. It needs a primer to begin with. So primase has to lay down a short segment of RNA base pairs to make an RNA primer. It's going to be temporary. You can't have a little sequence of RNA in your DNA gene. So that's going to be temporary. And then that RNA polymerase will start, okay, from that primer. Okay, let me draw the RNA polymerase right here. So this is the RNA polymerase. Directionality, confusing. Where the RNA polymerase is adding on to the strand, that is going to be the 3' end, because it's always adding on to the 3' hydroxyl group of the previous nucleotide. Okay, so it's adding those triphosphate nucleotides that have the energy with them. If this is the 3' end of the new DNA, then the other end would be 5'. Here's the old 3', and then here's the old 5'. Okay, so starting from, you can kind of work your way out if you had a question dealing with that. Again, this would be always adding on to the 3' end of the new DNA. What is keeping these two strands separate? There's an enzyme called helicase. We say that unzips the two strands. Okay, and there's some stabilizing proteins that kind of keep that open. And then the opposite strand, it's going to be working in a fashion kind of like this, and then like this, that RNA, that DNA polymerase, okay, in Okazaki fragments. Eventually, again, moving towards the replication fork, but so again, this is going to be 3' and there. So it's going to have a series of primers like here and here and here and here. Let me change colors here a second. So once though that RNA polymerase has done its job, another enzyme has to come in and get rid of the primers. So that's, sorry, not RNA, DNA polymerase 1 is going to, again, get rid of the primers. Okay, so we would, DNA polymerase 1 would come in here, again, fill in those primers. One more, what's the last enzyme that seals up the gaps? Ligase. I think I heard somebody say it. Okay, and then ligase, fill in the gaps. It actually adds a phosphate group that kind of, you know, around where the DNA polymerase 1 filled in. Okay, so again, you got to end up with a continuous strand of DNA. My artwork is really not quite on par with Khan Academy. Sorry about that. Okay, upstream here, one more enzyme to talk about, DNA gyrase. That's supercoils. It can get super twisting when we keep unzipping that DNA. So the gyrase makes cuts, unwinds it, and seals it back up again to alleviate that supercoiling. Okay. So I have a little video that I'll go ahead and post it that kind of does the basics of transcription and translation. So I will post that. If anybody wants to stay and has questions, I'm here. I'll be in my office for a while. I'll be in my office all Monday morning as well. So if you have questions, shoot me emails. Happy studying. Thanks for coming to class, you all.
Okay. Final electron receptors for